It's my pleasure to introduce Steve Pacala, who is now Emeritus, Frederick D. Petrie Professor of Biology, and also an HMEI Senior Scholar. Steve is known to many of us as a sort of a wonderful thinker, somebody we can learn a lot from, learn a lot with, studies climate, biodiversity, carbon, and the way these interact. And so we're going to get a treat of a wonderful science discussion, a way to think about the connection between biodiversity and climate. I could go through the very long list of honors, distinctions, appointments that Steve has had. But I think the couple that I will focus on is Steve was director of the Princeton Environmental Institute, which is a precursor to the High Meadows Environmental Institute. And Steve, initially along with Rob Socolow, was the co-director of the Carbon Mitigation Initiative and now remains as director of the Carbon Mitigation Initiative, is a member of the National Academy of Sciences, American Academy of Arts and Sciences, and the American Association of Advancement of Sciences fellow there. Steve is currently, in addition to doing great science and teaching us all about many things, is chairing the National Academy of Sciences, Engineering, and Medicine Committee on Accelerating Decarbonization of the U.S. Energy System, and simultaneously is a member of President Biden's Council Advisors on Science and Technology. So very much connected to policy, but today the talk is going to be on science, but I've been told that you could even ask about policy afterwards. So Steve, thank you.
Yeah, so thanks. I feel like I should apologize for not talking about climate and energy policy, because many of you know me in that context, and there is a broad interest, of course, in that problem. I've got a lot to say about that, but I actually am not going to apologize because I paid 51% of my salary so that I have something else to talk about, okay? And that's what I'm going to talk about today. I'm going to talk about some basic scientific work about the direct interaction between biodiversity and climate. And so I'm just going to launch into it so that we can get through. Oh, there it goes. Okay, so there's an outline here. I'm going to start by just introducing the problem, interactions between plant biodiversity and climate. And then models of vegetation dynamics that exchange matter, energy, and momentum with the atmosphere, a major component of Earth system models, all right? Because this is one of the primary determinants of climate, and it's one that biodiversity has some impact on. And we have actually no theory at all about how diversity in the traits that determine how plants interact with the atmosphere, exchange matter, energy, and momentum with it, how biodiversity in those traits is maintained, nor what the interaction might be between that diversity and the climate, and in particular, how it might shape climate change. So I'm going to talk about an effort to sort of make some headway on that problem. And it's just a beginning, I'll warn you. I do come to some conclusions at the end, but it's by no means anything with a bow tied around it that's all wrapped up. I'm going to talk a bit about a thing called the competition colonization model and amensalism, which will turn out to be prominent. And then I'm going to talk about 4 examples of recent studies I've been engaged with, mostly with a senior scholar here named Matteo Detto, and also with people with Jonathan Levine and people in his lab, in which some generalities emerge. And then I'll talk about implications. All right, so let's just get started. Now, it is obvious that climate affects biodiversity. Anybody with even a modicum of training can land in an airplane anywhere in the world, glance at the vegetation, and tell you what the climate is, all right? And it's highly deterministic and determinative. And you can also tell a person how many species roughly of plants there are within bounds, what the distribution of abundance of those species are, and so on. So there's an obvious indication that climate affects biodiversity. This is the so-called Whittaker diagram, which is and extremely simple-minded but useful descriptions of the climate dependence of what we call the biomes, different kinds of vegetation. And it shows how mean annual temperature and annual precipitation determine where tropical rainforest is, where temperate seasonal forest is, and so on. So it's really obvious that that interaction, that vegetation organizes itself depending on what the climate is, probably more than anything else. What's less obvious is how the biodiversity might impact the climate. It's going to do so through exchanges of matter, energy, and momentum with the atmosphere, if you also include within that greenhouse gases. This is one set of experiments. There have been a huge number of experiments that have planted different diversities of plant species and then looked at how good they are at taking up carbon from the atmosphere and storing it on the surface. This is one experiment that is the so-called Cedar Creek experiment, Dave Tillman and colleagues. And it shows how the ability to maintain carbon uptake through a drought depends on the number of species that are planted pre-drought, okay? And this one asymptotes at around 10 species, which is what they typically do if the environment is simple. But if the environment has many challenges, many axes of variability, many different kinds of weather and that sort of thing, there's no evidence, like in the image over here, of an asymptote. More species you add, the sort of better the performance is, all right? And so if you just look at the sort of numbers, it looks like biodiversity is sort of zeroth order, a zeroth order determinant of carbon storage and carbon uptake. It's not the only one, but it's one of the ones that's got to be in the room if you're going to deal with it. This is another example, and it's the other biggie, has to do with water. I've got this one because I actually know the data well. I was a minor author on this, but made sure that the data analysis I believed. This appeared, I don't know, 5 years ago in Nature. And it shows at basin scales, using anticovariance towers, how the ability of plants to maintain evapotranspiration through drops in rainfall, through rainfall droughts, is better. There is less variability if there are more species present in the basin. And it's because variability in hydrologic traits allows drought-adapted species to sort of keep evapotranspiring when everybody else is shut down. And it reduces then the drought feedbacks between the atmosphere and the land surface that actually impact humans negatively, all right? So this is another service. And this one is also, you know, it's big enough to actually be in the room, right? It's not just a frill. Now, I'm going to be talking a lot about models to try to come up with some sort of way to think quantitatively about the kinds of phenomena I just showed you. And if you're a community ecologist, which some of the people in the room are, these models are different than the ones you're used to. All of the ones that I'm going to be talking about have at base individual plants and lots of physiology about how that plant opens and closes its stomates, pulls CO2 from the atmosphere, returns water to the atmosphere, takes up water from the soil, all that sorts of things. So there's equations for photosynthesis, for evapotranspiration, for energy balances, for CO2 diffusion, radiative transfer, stomatal conductance, water uptake by roots. There's a lot of different pieces here. And if you sort of poll the data and ask how ubiquitous are these functional forms, the answer is they're amazingly ubiquitous, right? So plants seem to have settled on a few ways of doing things quite a long time ago, maybe when C3 and C4 photosynthesis were first developed. And they've sort of stuck with the algorithms, or the algorithms optimized early. But the parameter values in those functional forms are highly variable. And there's no real theory for how those parameters that control the exchanges between the vegetation and the atmosphere, how that variability is maintained, or how it's going to work. And so that's what I want to talk about here. So I'm going to start with models like this that are physiologically and parameter rich, but relatively well settled across the biosphere. And you don't just like assume vital rates like growth rates or death rates. You actually derive them from the carbon balance and the water balances and stuff created by all that stuff, All right, so there's a way to do that. And I want to tell a couple of tales of plant woe because it's going to be important later. So plants first came out of the ocean and colonized the land, higher plants, around 450 million years ago. And that's kind of late in the fossil record, if you think about it, given that there were multicellular organisms twice that far back, and there were green algae way further back than that. And plants are basically, the photosynthetic parts are green alga. So why did it take so long? And the answer was, it's really hard to deal with water. Hard to deal with water because the atmospheric pull, to try to pull the water out of the plant, is enormous. Plants are in the ocean evolved to bring CO2, their primary food, in through a moist membrane. And when they try to do that here, they expose that moist membrane to the atmosphere, which pulls the water out of them. And it's got to pull the water out of the soil, and that sets up a gradient, a potential gradient that creates enormous tension inside the plant. And this is a big gradient, right? So if we had a scuba tank here, and I inadvisedly took a wrench and knocked the valve off of it, it might kill some of you, and it would go through the wall. And that pressure gradient is the same magnitude as the one, but it's in the opposite direction, from where we are here to the inside of a plant during a typical drought. It's a great big pressure gradient like that. So it's not surprising that during droughts, plants are constantly failing. Their xylem is getting embolized by air rushing in from the outside. And when that happens, it kind of wrecks things. And so it's really expensive to try to armor the plant to keep it from doing that and to repair it if it does. A good example is that plants have secondary growth. Trees continually add new xylem because the old stuff just wears out, gets all damaged up, right? A plant like a bamboo that wasn't designed or hadn't evolved as a grass, not as a tree, giant bamboos that big around and 20 meters tall have to get rid of their entire stem every 7 to 10 years. Let the whole thing die and make a new one because the xylem wears out and they don't have the ability to do this. So this is really expensive. Second thing about being a land plant that is a tale of woe and that leads to big expense is that it turns out that being on top of the land as opposed to floating and mixing in a euphotic zone means that you can reliably shade your neighbor and your neighbor can reliably shade you. And this creates a Nash theoretic nasty race to the canopy that causes plants to invest in this extremely heavy and unproductive stem, mostly as a defensive and an offensive weapon. There are about 3 trillion canopy trees in the world, believe it or not, between 1 and 3 trillion, different estimates of different numbers. But each one started out as a race between something like 1,000 seedlings per square meter. And they're occupying 100 square meters. And so you get this 100,000-fold reduction in density along that race. So almost every plant in the world loses its life to this race if it's in a place where the vegetation can get tall. And so this is really expensive also. Getting access to light and getting access to water, big costs. Okay, now, if we take a model of a physiological plant, and we take a whole bunch of them, of these plants, and we compete them against one another. This is looking down, this is an ancient video, looking down on a model running, and this is actually a white pine and a hemlock forest, and they're interacting. And there are all these seedlings that are growing up, and they're competing with one another, and all this physiology is going on. And this is a very complicated object that Simon and I worked on for many years, and it's actually used, this one and others are used all over the world to manage timber, and people pass laws based on what they say. They actually work. But they're not so good for our purposes. We need a way, we can't simulate every tree in the world. It takes too much time. And so instead we have to derive what are known as macroscopic equations. So let's randomly toss a small box on the ground in a simulation at time t. Count the number of individuals of species I in it whose size is between S and S plus delta S. And if the box is small, that's always either 1 or 0. It's a Bernoulli random variable. And then repeat this a large number of times, calculate the mean, divide by the area, and what you have is the density of plants of species I and size S at time t. And that's the thing, just like in the atmosphere, where you want to iterate on the concentrations of gases, right, in the atmosphere. That's the thing that actually forms the basis of a population dynamic model. And so the trick for many years here, Simon's lab and my lab worked on this for a long time when we first came here, was to come up with a way to do that, to derive what those macroscopic equations were. And we eventually came up with two different ways of doing it. One's called the ecosystem demography model, and one's called the perfect plasticity approximation. And they work best together, and they work so well that we've all given up simulating the individual plants, because these sets of integral partial differential equations, which are tractable, allow you to know what the simulation is going to do using only the information about the individual plants before you ever run it, all right? So what I'm going to be talking about is the macroscopic equations that come from those, okay? Now, those macroscopic equations are the equations for the biosphere that is inside GFDL's land surface model that Elena Shevlyakova and Sergei Malyshev have built over many years. It's this thing called LM4. So LM4 actually does all this stuff and makes predictions about the exchange of matter, energy, and momentum with the atmosphere, and contains all this individual biology. You could actually interrogate that model and say, what should the size distribution of trees on Barrow, Colorado Island be in Panama? And the model will actually tell you, and you can check it, all right? And incidentally, it does a really good job of that. But the point is, this is an object that comes from that tradition. And so it's suited in a way to ask this biodiversity question. What happens if you had a huge amount of diversity in all the parameters and allowed the plants to compete with one another? But it turns out this is so complicated to solve numerically, it's so expensive, that it's really difficult to contemplate a research program where one tried to do that. It's not possible to run a model for long enough to find out which species is going to win if there's a huge number of species. It's not possible you can't afford to do all of the simulations you'd need to do. And so what's done instead is there's very restricted biodiversity in one of these models. These things compete with one another locally, but there's so few that you always have one winner. And that's the state of the art here. The emerging state of the art is to impose distributions of parameter values that represent the biodiversity of those parameter values on this thing, in the same way that you might impose distributions of sea surface temperatures on an atmospheric model. And it works in the same way that prescribed SSTs do, but also with the same limitations. You limit how the system can interact with itself and limit what it'll predict under climate change. So we were looking for a way to do this interactively by taking versions of this thing that retained its essence, but also that were simplified in ways that were sensible, like the weather is not variable for one day to the next, for instance, but there's still all the weather in there. There's still all the processes that go on, where we might make some progress using a pencil and paper and with mathematics. And so before I turn to the 4 examples of that effort, I want to introduce 2 other things, competition colonization model and amensalism. Now, many of you have seen this picture before. It's one that I like. You may not know, but Eno Hall was the center of what I call the heroic age of theoretical ecology. This is Lord Robert May, who's now dead, Robert MacArthur, who's now dead, and Simon Levin, who's sitting right there, okay? And if you know these people or knew them, the facial expressions really tell you an awful, I selected these with great care, okay? So I'm not gonna go through that, but if anyone wants to ask me later, I can't. But you know, I've never resolved who that is until this lecture, digitally enhanced. It turns out it's Henry Horn. Now, Henry Horn, as you know, died tragically in this very building 5 years ago next month, okay? And Henry was prescient, all right? He was a prescient sort of person. I have 2 examples of that. The first is mildly politically incorrect. Henry one time said to me when I first got to Princeton, Too much theory and too few muddy boots makes Eno Hall a mental institution, okay, with the double entendre about mental institution and theory being very much real. And of course, the university has now acknowledged Henry's position by having converted Eno Hall into its mental health center for counseling and psychological services. And if you look at the website, the architect says it's going to have soothing colors. And we could have used soothing colors, and we didn't have them. But it was a marvelous mental institution to work in. The other piece of prescience is this thing here. It's a patch dynamics model that's called the competition colonization model. It's very much a toy, but it's a toy that inside, unless you're a community ecologist, you sort of never heard of it, all right? And there was a paper in 1971 by Richard Levins and David Culver, and then Henry Horn and Robert MacArthur in 72 wrote the second paper on this model. It's a toy. Don't read either of these papers. They are dated, all right? It's like deadly trying to get through them, okay? Now, the competition colonization model is extremely simple. The version that's written up usually is in continuous time. I'm going to give you one in discrete time, but I want you to understand it. And so this discrete time model is the easiest to understand. We're going to randomly scatter seeds of annual plants on a grid of unit area cells. You can think of them as pots or something. Randomly broadcast them. If present within a cell, we're assuming that species 1 will kill all the species 2 individuals. Species 2 will kill species 3, species 4, species 5. So there's a competitive hierarchy. The lowest numbered species wins, takes the whole thing. If there are 2 of the lowest numbered species present, the fecundity of the cell remains the same. Okay, so either one kills the other or they share. And that's actually consistent with the way these plants operate. Per cell fecundities are in the reverse order. So the most competitive species has the lowest per cell fecundity. All right, that's it. Some definitions. If Ni of t is species size, population density, and generation t, and Si of t is the sum from species 1 through species I of those densities. So it's the cumulative density of the most serious competitors from species I through species 1. And again, the seeds are scattered randomly. So the seeds per cell is governed by a multivariate Poisson distribution, which means the fraction of cells containing 0 seeds of species 1 through I is e to the minus SIT, e to the minus sum. That's the 0 term of a Poisson distribution. And then we end up with this simple recursion. Population density this year is the fecundity of a cell of species I, is the fecundity of a cell of species I, times this difference between exponentials. And those differences between exponentials mean the following. If this box represents 100% of cells, this term, the 0 of a Poisson distribution, represents this fraction of those cells because it's all cells not colonized by a competitor superior to species I, which is the sum of the e to the minus density of everything from 1 to I minus 1. And this one is these cells containing only weaker competitors than species I. So this thing is the fraction, if you want, of resources that aren't colonized, aren't captured by stronger competitors. And this is the fraction that aren't captured by weaker competitors, and the difference is the fraction captured by species I, right? That's what it is. Now, what's weird about the competition colonization model is that it's got extremely complicated behavior from an extremely simple toy. Repeated invasions of randomly drawn Fs, right? F sub i's. And remember, they're in order, so that shows where they are in the competitive hierarchy. Causes some to fail because there's a limiting similarity. It's too close to one of the others. But some succeed. And on net, the diversity climbs as you keep throwing random Fs at this thing. And as it climbs, weirdly, the limiting similarity itself collapses in such a way that in the limit, you go to infinite diversity. This thing will pack and maintain an infinite amount of species diversity. It does so at an interesting mathematical limit, whereas if you look at the density of n of f, in the continuous limit, it's continuous, I believe, Simon, but not differentiable. Remember that? It's like infinitely jaggedy thing, all right? So it's a cool object that it goes to. And subsequent removal of species, once it reaches high density, typically causes a cascade of extinctions after a period that Dave Tillman talked about as an extinction debt, and something that we actually should worry, conservation biologists should worry about. Randomly pulling a species out could lead to a cascade of extinctions, he said, because of this. This has been much criticized because of the assumption, all of the criticism is that it assumes what I'm calling hierarchical amensalism. And what is that? Amensalism means that species A has a competitive effect on species B, but species B has no effect whatsoever on species A. If you look this up in Wikipedia, they'll give you all kinds of examples. If you look this up in ecology textbooks, they'll give you all kinds of examples. And if you think about those examples, they're terrible because they're not clearly amensalism at all. It's not obvious that what they say is true. It's really difficult to find good examples of amensalistic interactions in nature, let alone ones that are hierarchically amensalistic. And by hierarchically amensalistic, I mean that species one, abundance, change in abundance, population dynamics depend only on its own density. Species two depends on 1 and 2. Species 3 depends on 1, 2, and 3, and so on, okay? So that each species has a competitive effect from lower ranked species, 1 through lower numbered species, and no competitive effect of ones that have higher numbers than it does. And it's in a perfect hierarchy this way, all right? They're very difficult to come up with unambiguous examples of either of these, let alone hierarchical amensalism. But do see, if you're going to look at one paper on this, there's a paper by Lindsay Turble, Mark Rees, and Mick Crawley, which is a sowing experiment in this journal you can look up on the website, where it actually looks like that might be going on in chalk grasslands in the UK. There are tons of theoretical studies that show that amensalism in general, and this is just 3 of them, stabilizes ecological communities. If you put a little bit of amensalism into an ecological community, it stabilizes the whole network and really promotes biodiversity. It creates compartmentalization that is really useful for things to hang together. And there are even some recent examples, this one from the latest PNAS, of hierarchical amensalism being even better at promoting diversity. So we know that this mechanism, if it existed in nature, would be really powerful at maintaining diversity. Okay, so let's look at some examples. The first one is close to a toy model. I'm going to put her on the spot. This is Ruby On's work, and it's just a tiny little nugget of Ruby On's very, very interesting theoretical work on how shrubs are invading the Arctic and why, which has serious climate implications. And so we've got this little model that's at the core of something that becomes grander, but I want to just tell you how it works. Most of the plants up there are vegetatively reproducing, which means they produce copies of themselves vegetatively, all right, from stolons. Plants, we're assuming in this toy, have the same crown area, but differ in their height and thus mass, so that there are tall plants there and there are short plants there, and this is actually true, right? Plants in this model gather carbon throughout the growing season and then deploy the new adult-sized clones at the beginning of the next season. All right, so they gather their carbon, they wait till the winter's over, and then they say, here's the new ones, right? And the number of offspring is proportional to the carbon captured, which is proportional to its crown area, which is the same for everyone, divided by the mass of them, right? So you have, if they weigh twice as much and you have the same amount of carbon, you can only make half as many, right? So that's the way this thing works. Photosynthetic rate is proportional to crown area and downwelling photon flux. This is not crazy. Photosynthesis has a light reaction where it depends on photosynthetic flux, the photosynthetic rate, and then it levels off because it becomes dark reaction limited, right? So Rubisco determines how much CO2 you can fix with that energy. But if the plant upregulates Rubisco and makes more of it, it can extend this thing, this kink point up, and if it adds even more Rubisco, it can do this, and plants can move their Rubisco up and down. And there's quite a bit of evidence that they do so to put the kink point where the light is, all right? Because it's inefficient to have a bunch of Rubisco that you aren't using around when you can use it for something else. And so light in this model is governed by the very simple Beer's law, which is an exponential decay where what's in the exponent is the leaf area above. So light decays exponentially using leaves as a neutral density filter. The leaves above determine how low the light level is. So it's possible to take all these physiological parameters and submodels and everything and go to work on them. And by going to work on them, you want to reduce them to the simplest possible form, which means that what I call a canonical form, where the number of parameters that you have left over is at a minimum, but it's minimized in such a way that it doesn't take any of the mathematical behavior away from the system. So that if a whole bunch of parameters always occur in the same sort of algebraic cluster, you just rename that as one parameter. You're allowed to change the time scales and the spatial scales at will so that a parameter disappears. So that, and you can be pretty ruthless about that. You can normalize the density of each species using different area, species-specific area metrics. But so be advised that everything I'm going to show you that's an equation, to keep it simple, is in this kind of canonical form, but that the mathematics hasn't been destroyed, okay? Biological interpretation means you have to re-unpack it, right? Okay, so when the smoke clears, this is how simple that model becomes, this thing here. Population density change is equal to this object here. And if you compare that to that's the light beneath taller species, and that's the light between smaller species, and the difference is the light absorbed by the canopy of species I, and carbon gain is proportional to that because of the kink business I was talking about. And if you compare that to the competition colonization model, it's exactly the same. Except that this D here, which is a death rate between years, if that were 1, it would be exactly the same. Or if you made this an equilibrium, where this term equals that one, this term equals that one, the D gets folded into the F's. And the equilibria are the same. All the models I'm showing you, the equilibria determine all the behavior. So if you know the equilibrium exists, you know that all the species that have positive abundances can invade when rare. You know everything, all right? And so this model and this one turn out to be the same, even though the light model has nothing to do with the processes that were in this competition model, which was all about, you know, capturing space and stuff like that and excluding things. Nobody's dying here. They're just shading each other, and they're all perfectly well alive and everything. And yet it's identical, which means it's a mathematically identical behavior. Same bizarre dynamics. Infinite limit, collapsing, limiting similarity, all that stuff. Somehow this thing emerged. I don't know of another example where the competition colonization model has just emerged, in this case from plant physiology, right? Now, I'm going to turn now to an example we published 3 years ago. Teodato here is the first author. Jonathan Levine, who interactions with him really got this all started in my head, and I want to thank him for coming to Princeton because of that, and other things too, but that one in particular. And we published this thing, and this is the full enchilada, all right? This is a full-sized forest simulator with weather that is relatively constant, but everything else, all the complexity is there. All these guys are competing against one another, macroscopic equations, integral partial differential equations. You go into it and you think there is no way I can possibly solve this thing. And somehow everything falls away and you can. And so we actually figured out how this thing works. Even though there are, you know, scores, dozens of species-specific parameters, there are actually only 2 that determine everything. One of them is the time to lifetime reproductive success 1 in full sun. After a disturbance, a bunch of little plants are sitting there, and you say, how long do they have to grow in full sun before the babies they make can survive the coming period of darkness and end up in another disturbance somewhere and end up at the same place you started? And when does that expectation reach 1? How long do you have to grow before that expectation reaches 1? And that differs between the species. And it depends. The more you invest in a stem, for instance, the longer that takes, because you're investing in stem wood to make yourself tall, which doesn't allow you to produce a lot of babies in full sun. But the other one is the height growth rate. So if you take off species-specific parameters and get a scatter going, then there's a little algorithm where you take the tallest one, the next tallest, the next tallest. Each time, you have to run a little, you can actually do this on your phone, okay? They're a big complicated formula, but on the calculator on your phone, you can figure out from 40 species-specific parameters what those two values are on this graph, and figure out whether or not, what those values are, and use this algorithm to figure out who's gonna win. And then there's a little recursive step that you need to do with a tiny little program that says, no, I'm too close to a neighbor, all right? And so that one's too close. And all the others aren't gonna coexist. And so coexisting species inevitably show a trade-off between height growth rate and the rate of accumulation of lifetime reproductive success. And so we're gonna order them 1, 2, 3, 4, 5, okay, just like before. And so this is the thing that complicated simulator is gonna converge to. If there's a simple trade-off, if you have variation in a single parameter, like say allocation to STEM, it will oftentimes create a monotonically increasing trade-off in this space. And in that case, there will be an infinite number of species that coexist in the limit, although there will be some limiting similarity and stuff. So this model is not mechanistically amensalistic because all species of plants shade the young plants in the understory and inhibit their growth and promote their death. But all these models also have that terrible race to the canopy I was talking about, where 100,000 start and you only end up with one that survive. And that race turns out to so dominate the process that it becomes effectively amensalistic, and hierarchically so. The forest model looks nothing like the competition colonization model. It's possible to write down in canonical form these equilibrium equations showing where that complicated simulator is going to settle down. And if you look at it relative to the competition colonization model, it's almost the same. There's one little difference here. This 1 over theta is about 2/3. So this is 1 2/3 as opposed to 1. And then there's this SI minus 1 to the minus 1 over theta thing instead of an e to the minus thing in there. But those two functions, to the minus 1 over theta and the e thing, are just two concave up functions. That's almost the same too, all right? So these are just two simple little concave up functions of these S sub i's and S sub I minus 1s. And so these two models, it turns out, are almost identical, even though the one is a toy and the other one started out as this complicated thing. They have the same qualitative behavior as a result. Same infinite diversity from successful invasions, same excuse me, same declining limiting similarity, the same extinction debts, and for the same reason. All right, if you start with the total number of photons arriving between disturbances, this term is the photon captured by species I and a weaker species. This term is the photon captured only by weaker species. The difference is the photons captured by species I. And so what this is both the light competition models and the competition colonization model are mathematical variants of the same hierarchically amensalistic model of resource competition. They're actually the same thing. They're just two different variants of the same thing, of what happens when you compete for resources and have this hierarchical amensalism embedded in it. This was a surprise. Again, we didn't put this in there. This emerged, all right? We thought this might be due to the directional nature of light, that the amensalism was due to directional nature of light, right? So taller species shade shorter ones, but not vice versa. And so we shifted to water. And this work is with Jacob Levine, Jonathan Levine, and me. Jacob has done a whole thesis on another part of this problem, but a lot of these slides even are his. So he's really the driver behind this. Plants in this little simple example I'll show you are dormant for part of the year. There's constant rain during the growing season once they wake up. These are the plants. They have gas exchange, photosynthesis, and stomatal conductance. When they open to breathe CO2 in, water escapes. There's soil moisture uptake by roots and so on. It's all the same things we talked about before. And when the soil dries, the water potentials, the tension inside the plant xylem and inside its leaves go up. and eventually would cause them harm. So they have to shut down when that gets too high. Just like, remember the scuba tank example. So this is the way that actually looks. That's a stomate. And when there's too much tension inside the xylem, right, when the pressures get really low inside the xylem, these cells lose their water and they close, right? So they close and they stop the water from escaping, stop the CO2 from coming in. So you shut down the photosynthesis. And the switch that does that looks like this backwards sigmoid curve, where that's water potential. But water potential units are useful if you're interested in free energy, but plants are interested in how much water there is volumetrically or mass of water. They want to know, they have a certain mass amount of water they can use, okay? And when they do this, what you end up with then is change units to volumetric soil water content. This thing becomes a square wave, and plants shut down at a species-specific water level, volumetric water level in the soil. So plants photosynthesize flat out, they reach a species-specific level, and then they turn off, okay? So low water potentials, being able to photosynthesize at low water potentials, it was really costly. Requires thick-walled xylem, deep roots, all kinds of things. And so we can set this up as a problem and do the same thing we've done before. There are two examples I've got. One uses the simplifying assumption that the water levels in the soil are at quasi-equilibrium, meaning the rain is balanced by planned uptake and purely physical losses. And the other case is going to be dominated by transients. Make a long story short, didn't make a darn bit of difference. You still end up, this thing is identical to the light competition model. And it's, again, hierarchical amensalism, even though the water isn't directional. If you want to ask in the Q&A, I can tell you why this seemingly non-directional pulling water from the soil is the same as directional light shading. So you get this thing again, emerges again, all right? Last example is from Jacob Levine's thesis. He's done a lot of empirical work showing that this is correct. This is the opposite bookend from the quasi-equilibrium water, where winter rains recharge a soil, then the rains stop, the plants germinate, and they mine water that's already been deposited until it's gone, and then they stop. This is what you see in a California Mediterranean grassland, where they all, he and Jonathan work. So to make a long story short, getting back to canonical form with this thing, all of a sudden finally gives us something else. What does it give us? It gives us a set of terms here that, if you're a community ecologist, looks very much like a traditional model. The difference between the water content at the end of the rains and when the first species shuts down is divided among, in this case, the 3 species in the model. If there were 4 species, you'd have a thing that looked the same, but with 4 equations in it and an extra term, right? 5 would look the same again. So this thing is a familiar, what's called a Bond-Verdelampi equations for the scramble competition, and there's one winner predicted in this thing, and it's going to be species 3. This thing is a hierarchical amensalism model that's added, and it's because only this species has strong enough xylem to mine water between its shutoff and the shutoff for the next most armored species. And so you're adding hierarchical amensalism to a traditional ecological model. And because I'm out of time here, to make a long story short there, this partial hierarchical amensalism is an extremely strong promoter of diversity also maintains infinite diversity without any limiting similarity and without any extinction debt and extinction cascades. And so this partial hierarchical amensalism, I'm calling a model in which we have terms. The last term in each equation looks like hierarchical amensalism, and the first one, everybody competes with everybody else. In this case, all the species, the drought tolerant and the intolerant ones, can all compete for water when it's nice and wet right after the rains quit, but at the end, there's only one standing at the end, okay? So I suspect that partial hierarchical mensalism will be quite common and should occur whenever a resource is divided by biological or physical factors along an axis separating easy exploitation from expensive exploitation. All right, I think this is going to be real common. And I had a lot of examples in a longer talk that I'm not going to show you. Coexisting species, if this is operating, will thus seem to differ in what you might think of as diet breadth. Those with broad diets are able to eat both difficult and easy resources and will have low R0s, R0 in the COVID sense, right, reproductive number, because of the investment needed to handle the difficult resources. Species with a narrow diets will only consume the easy resources and have high R0. So the conclusions from the talk are that interspecific variation among plant traits likely to affect climate leads to complete or partial hierarchical amensalism. This was a real surprise to me. And that then promotes diversity very strongly, probably in nature and in models, right? Surprisingly, the mathematics of the obscure competition colonization model emerges from mechanisms of competition among plants for light and water, which have nothing to do with mechanisms assumed in the original. The partial hierarchical amensalism, I predict, will be common in nature whenever a resource is arrayed along an axis separating expensive resource capture from inexpensive exploitation. And finally, partial hierarchical amensalism, like full hierarchical amensalism, strongly promotes biodiversity in all the examples I've looked at, even better than regular amensalism. But if you're a theory person, this is actually a good problem, all right, because it's not been done in general. On the climate side, partial or full hierarchical amensalism will cause coexistence of many functional types in models like LM4, big capital, unless any practical numerical scheme is too coarse to represent the critical dynamical differences among the types. So now we know what to look for, and if it doesn't happen, there are only a couple of candidates, and the most likely is that the numerical package is not working the way it needs to yet, okay? Interspecific variation among plant traits, I predict in general will increase carbon storage. Knowing how this hierarchical amensalism works, I think it's going to be a big deal. It's like 50% of the carbon stored on the planet will be because of the biodiversity maintained by this hierarchical amensalism. And it'll have a significant impact of anthropogenic emissions on climate. And interspecific variation among plant traits will also reduce modeled drought for the same reason it does so in real ecosystems. And this will also be a sort of a zeroth order part of that feedback that also limits the impact of anthropogenic CO2 emissions on environmental damages. And that's it.
All right. Well, thank you, Steve. I'm going to note Simon's question, but as we've been doing, we would like the first question to be from a student or a postdoc. Simon, yes.
So in two cases of light and water.
Oh, wait, let's wait for the microphone because we have a Zoom audience, and I'm going to put on my glasses and look for questions from them after Simon's.
In these two cases of light and water competition, what would happen if you have multiple resources being competed for at the same time? Should we think about this as a linear superposition, or once nonlinearities come into the picture, how does the emergence of amensalism, how do you think about that?
That is a great question, all right? And it's one that that Jacob and Jonathan, we were actually talking about this today. And we actually have work in progress that's aimed at that. The real interesting question is when, you've got variation among 40 parameters that control the uptake of water and the loss of water, and that also determine the uptake of light and the uptake of carbon and the storage of carbon, right? So collectively, they do these things. And these mechanisms are just emerge from the competition for those things. And so it ought to be that in systems in which light and water are simultaneously limiting, like they are like in every Western forest, right? The plants compete with one another for light, but also there's drought during every summer where they're competing for water. It ought to be that both mechanisms are operating. And the interesting question is, are they additive in some sense, or are they not? So do they make coexistence somehow stronger and buffer the communities from biodiversity loss better or worse? And so that's something I'm very much interested in knowing, because a lot of other phenomena that have been observed in nature would hinge on the answer. So great question.
Okay, so now we'll have a question from Simon, if we can get the mic. And
Okay, wonderful as always, Steve, thank you. The equations from which the infinite diversity arises are all deterministic. And it would seem that if you added a stochastic term, it would go away. And you ought to be able to, from the mechanistic models, to quantify what the uncertainty, the stochasticity is.
Yeah, so there's different kinds of stochasticity in a model like this. The macroscopic equations, of course, exist and work because of the underlying stochastic process that exists there. But I think you're talking about some sort of environmental stochasticity on top that would cause
Or
Lyme.
demographic stochasticity.
Because of what?
Demographic stochasticity.
Well, demographic stochasticity these models include. But so the macroscopic equations are about the means over a stochastic process that has tons of demographic stochasticity, and it's that demographic stochasticity in the underlying stochastic process that is maintaining the diversity to a great extent. But your question is correct in that as you add biological complexity to these models, you'll notice that, for instance, in Ruby's simple toy model, we assume that the taller species shades the smaller one completely and the smaller one shades that. Well, really, their canopies overlap a bit, okay? And so when you include all that correctly, you'll get very high diversity, but you lose oftentimes the limit of infinite diversity, which is an annoying feature of these models anyway. You still get extremely high diversity that's robustly maintained.
Sure, I believe that.
Yeah.
Can you quantify the degree of environmental stochasticity that would go into the underlying mechanistic model?
Yeah, we in Mateo's ecological monograph, we do that, okay? And we use the example where, if you've ever been a gardener and you try to plant trees or shrubs or something, a lot of times some of them take off and some of them look terrible. And oftentimes I find that if like a holly has seeded itself and has already sort of survived, you should leave it alone because it's really going to grow well. And I think there's real difference in quality among individuals. I think a lot of it may have to do with how sick they've gotten and what kinds of parasite or pathogen loads they're carrying. And so that's what we actually modeled, and we have a whole treatment of that.
so let's have Lars, then Lore, keeping track of hands.
Hello. Yeah. Thank you, Steve. That was a summary of decades of thinking and development. Very nice to see. I'm wondering if you can tell us a little bit more about the equilibrium assumption and how it plays out. I think it's a quasi-equilibrium at the landscape scale, but at the same time, there's a time dependence. If you have two species or three species that compete, there's time dependence between the best species and extinguishing the other species. So this has to do with succession, right? And so to what degree is the biodiversity that emerges linked to time optima and linked to the decay in outcompeting your neighbor? And to what degree is it independent of that?
So what in a talk this short, you don't have time to say everything. The forest model, which is the one that is sort of most relevant to the climate question. What that model is of actually is not just one patch of forest where all the trees are competing and all that stuff is going on. It's actually a mosaic of such patches that are being disturbed and are in different stages of recovery. And so one of the reasons why it's so surprising to be able to handle that is you actually have to be able to solve for the nonlinear, time-dependent recovery equations for the recovery following disturbance to be able to solve for the equilibrium distribution of carbon in the landscape as a whole, okay? And so it's actually the latter that we're dealing with. And it's precisely what you said. It's the successional recovery that is maintaining the diversity in that mosaic, that landscape mosaic. But it's at equilibrium at a much larger scale. And if you were on the ground, it would not look like equilibrium, like the tornadoes coming and knocking everything down, right? Yeah.
Laura?
Yeah, so I was curious to know if that's, so like, is that what they're gonna, starting to implement in those climate models, those ESMs? What's the time scale for seeing those type of formulations versus what we have now?
Well, Matteo's group is doing a bit of this now. And in my view, the work needs, and it really, the question, it depends on what we find out, okay? I'm convinced that the mechanisms that we've got here are the ones that are going to be operating in those models. There might be some extra ones that come from the extreme variability of the weather. And I know that the numerical coarseness that is tolerated by a model that doesn't have to maintain diversity is not going to be sufficient for one that does. And so we're going to have to actually hunt that down. And it's a question of what we find out. It could be that we'll discover that the computational burden is still too strong, and we're going to need an NVIDIA, emulator, all right, to be able to do this.
Okay, and so we will have our final question from Frederick, as we do.
Is there no long-range spatial or temporal communication between these actors, given that now they scatter seeds and it's a hit or miss, or they shade or it's on or off, but isn't there any chemical communication through roots or rhizomes or through time where a seed can sit there for a couple of years before actually springing into action, that sort of thing?
Yeah, so there are multiple mechanisms that will couple dynamics. There are exogenous ones where large disturbances can couple the dynamics of large areas. You're talking about something else, though. The forces that sort of draw them back together, the simplest one is seed dispersal, which in our model can be really pretty long range, right? The chemical communication among plants is also very interesting, but that typically operates over tens of meters, maybe 100 meters, all right, but not scales of kilometers. It's known, for instance, that with plants, if you damage their leaves, they release low molecular weight organics like isoprene that leaks out of their leaves, and the other plants chemically detect it and upregulate defenses because they know somebody's being damaged around them. I actually had undergraduates that did experiments like this. It was really fascinating. You could show that they pass. In one case, the plants, she showed definitively that the plants that were damaged passed the information through mycorrhizal symbionts, okay, to tell the other plant
But this is not necessary to incorporate into your modeling because it wouldn't have a dynamic effect on?
We don't even have herbivores in this model. We don't have herbivory, period. Herbivory, sadly, unless you're an herbivory person in most ecosystems, is kind of second order. I hate to say it, all right? But it's only a few % of leaf matter gets eaten annually, whereas 100% of it in a deciduous system dies annually.
But maybe not for the herbivores, is it second order?
Yeah, I had a whole bunch of really good examples of one of the examples of partial hierarchical amensilism is I think most grazing systems are separated between herbivores that can only eat really luscious little plants, like an African deep, and great big-bodied mowers that can eat really coarse grasses. But every time they get a succulent herb, they eat that too. And if you think about that system, that's a perfect partial hierarchical amensilism. You got a broad diet among the slow thing, and you got a really restricted diet among the other. And I think this mechanism is actually going to be everywhere.
Well, let's thank Steve again. And thank you all, and remind you that on March 5th, our next speaker will be Catherine Peters from Civil Environmental Engineering.